Right, we'll run through everything you need to know about the immune system for the FRCA primary. So your immune system is your defense against infection, and you have two types, innate and acquired. The innate immune system is fast, non-specific, and you don't have to have had previous exposure to the organism. The acquired immune response is slow, specific, and requires previous exposure for it to work. And your acquired immune system, your acquired immune response, can be further divided into cellular and humoral. So when you get an injury or exposure to an invading pathogen like a bacteria, your innate immune system is the first one to activate. The idea is that these work in tandem, so your innate immune system would fire up first, allowing time for your acquired immune response to activate, and between the two of them, they destroy the invading pathogen. So you often get asked, what are the different components of the innate immune response or the innate immune system? And there are three main things. First of all, you have your barriers. So you've got your skin and your mucosa. Those have got a flora of bacteria on them, and those act as the first line of defense, preventing microorganisms from getting in the first place. If a microorganism does manage to breach these barriers and get into the tissue, there are then tissue bactericides, which are things like lysosomes containing enzymes or the complement system, and they essentially are like grenades waiting to explode and destroy whatever breaks in. And if that doesn't work, you've got leukocytes and macrophages, white blood cells sitting there ready to go, and they can have a phagocytic response, so they literally engulf and destroy any invading pathogens. And then on top of that, you've got a more widespread inflammatory response, which generally makes the tissues hotter and more acidotic, more difficult for the pathogens to survive and reproduce in. Now, this inflammatory response, much like many processes in the body, is a cascade system, the idea being that each level of the cascade sets off multiple agents at the next level, and you get this huge amplification of an originally very small signal can produce a huge response by lots of downstream activation. So an example of this process is bradykinin. So you have an enzyme called kallikrein, which converts kininogen into bradykinin. And you also get activation of cyclooxygenase 2, which converts arachidonic acid into the eicosanoids. And the eicosanoid that you'll probably have heard of is prostaglandin. Now, these proteins are like signals that are released by damaged tissue whenever a bacteria invades. They can also be released by specialized white cells like mast cells and macrophages, and these will also release things like tumor necrosis factor alpha, interleukins, and histamine. And altogether, these pro-inflammatory cytokines trigger an inflammatory response, so they vasodilate the blood vessels in the area. That causes an increase in heat delivery to the area. It allows white cells and fluid and other components of the immune system to flood into the area, and it makes the area hot, painful, red and swollen, all the features of an inflammatory response. And the idea is it makes it very difficult for the pathogen to survive in that environment. If you get asked about white blood cells, the ones you really need to know about are neutrophils, lymphocytes, monocytes, and then you've got your basophils and your eosinophils. So most of your white blood cells are neutrophils, and these are generally phagocytic cells that are involved in that innate immune response, and they also release lots of inflammatory mediators as well. Lymphocytes are generally more involved in the acquired immune system, apart from natural killer cells. So these are lymphocytes, but they're more innate immune response. Eosinophils are involved with parasites and worms, weird, wonderful things like that. And also, if you've got autoimmune disease, you often get a rise in eosinophils. Basophils, no one really knows what they do, but they seem to be the circulatory equivalent of mast cells. And monocytes are like macrophages, but in the blood. So acquired immunity or adaptive immunity is much more specific and targeted, and it requires previous exposure. So what happens is the immune system recognizes certain antigens or flags on invading microorganisms and produces antibodies or immunoglobulins against those antigens. And like we mentioned, there's cellular immunity and humoral immunity, and both of these are forms of acquired or adaptive immune response. So if you get asked about the cellular adaptive immune response, we're generally talking about T lymphocytes. They're called lymphocytes because they're found in the lymphoid organs, and they sort of circulate between blood and lymph, and they're constantly on patrol looking for these antigens. And each lymphocyte can only recognize one antigen, and that's why you have hundreds of millions of different types, because they can each only recognize one particular antigen. So you have to have lots of different types for it to be of any use. So what essentially happens is something invades the body, like a microorganism, a bacteria, a phagocyte, like a macrophage, sitting in the tissue where it's broken into, will engulf and phagocytose that invading organism, and it'll essentially rip it apart, grab all the antigens it can, and present those on its cell surface membrane for these lymphocytes to then come and examine, and they present it using what's called the MHC, the major histocompatibility complex. That's the machinery that they have that allows them to present these antigens to the cells that need to be able to see them. So the helper T cells come along, they see the MHC holding out this antigen, and that triggers them to release inflammatory cytokines, and that recruits B lymphocytes and other macrophages and other white cells to come along and help. And then the cytotoxic T cells will come along and destroy any cells infected with microorganisms. And the key point here is the activation of the B lymphocytes is what then leads to your humoral responses. These are the ones that can make antibodies. So when an antibody is produced, it's essentially a little fragment of protein that binds to the antigen, and it can do one of several things. It can inactivate the antigen and cause the cell to stop functioning properly, or it can act as a flag for another phagocyte to come along and destroy the bacteria. And it can also be an opsonin, which means it can activate the complement system, which we'll talk about next. In terms of types of antibody, there are 5 you need to know about: G, A, M, D, E. G is the most common, and an antibody molecule has got 4 sections to it: 2 heavy chains, 2 light chains, linked by disulfide bonds, and the light chain has the variable region, which is tailored to the specific antigen that it's going to bind. So on first presentation, it can take 5 or more days to produce meaningful amounts of antibody against a pathogen. But if you've had exposure beforehand and you get a repeat exposure, it only takes 24 to 48 hours to get a massive upregulation of production of the necessary immunoglobulins or antibodies. So that's why you have your innate system, which does the first 24 hours of trying to keep things under control, while the acquired system revs up and gets up to speed, and then that does the more targeted damage to the individual bacteria. So lastly, the complement system, you don't need to know this in very much detail. You just need to know that it's a bunch of proteins in the serum that work as a cascade, and together they form a membrane attack complex, which then physically destroys the membrane of the invading pathogen. It has 2 pathways, the classical and the alternative. And you start with C3, then you get C5, and that binds with 6, 7, 8, and 9. And all of these together form the membrane attack complex. And that's pretty much all the detail you need for the FRCA.